Hey guys, welcome to Chapter 12, the nervous system. Welcome to our new unit. So for today, we're going to name the major divisions of the nervous system, and we divide it both anatomically and functionally. We're going to describe the functional and structural differences between gray matter and white matter structures. We're going to name the parts of the multipolar neuron in order of polarity. We're going to list the types of the glial cells and assign each one to the proper division of the nervous system along with their functions. We're going to distinguish the major functions of the nervous system: sensation, integration, and response. We're going to describe the components of the membrane that establish the resting membrane potential. We're going to describe the changes that occur to the membrane that result in the action potential. That should remind you of the action potentials that we've already discussed. We're going to explain the differences between types of graded potentials and categorize the major neurotransmitters by chemical type and effect. So we're going to begin with a case study. Okay. So this is a brain versus spinal cord case study. And if you're a little bit older like myself, then you may remember the show ER, and this comes from that show. So Dr. Green and Dr. Carter were nearing the end of the first year of their medical residency in the emergency department of County General Hospital. It had been a long year and a long week, and they had been on duty for the last 12 hours. Things were not slowing down. "What are your plans for the weekend, Ken?" Dr. Green asked. "Sleep. What else?" Dr. Carter replied. "I hope things slow down a bit," Dr. Green said to himself. Suddenly, a call came over the radio. Paramedics were bringing in a young man with injuries sustained in a diving accident. A minute later, the doors to the department burst open, and the paramedics wheeled in a young man. "What gives?" Dr. Green asked. The senior paramedic, Jim Morrison, reported that the patient was swimming at the local quarry and did a forward flip into the water, striking some submerged rocks. "Which part of his body struck the rocks?" asked Dr. Carter. "He was in a hyperflexed, tucked position when he hit the rocks." Okay, so that means that his head was sort of tucked forward. Okay, he lacerated the right side of his head and neck and upper back. The patient indicated that he had severe pain upon impact and loss of sensation and movement in his arm and leg. He may have lost consciousness, but he's not sure. He also complains of a severe headache, dizziness, and nausea. When we arrived at the scene, we immobilized, stabilized, and transported him immediately, Jim explained. Looking at the patient, Jim continued. His name is Mike Smith, and he is 22 years of age. His vital signs include slightly lowered blood pressure, 100/70, heart rate of 75 beats per minute, respiration rate was normal, and he is conscious and alert. As the patient was being prepped for examination, Dr. Carter and Dr. Green discussed how they should approach their evaluation. Dr. Carter started by saying, "After seeing the head wound and the amount of blood loss and hearing his complaints, I want to confirm my suspicion that this patient has a brain injury." Dr. Green disagreed. "I think that the other signs and symptoms indicate a spinal cord injury, and that's what we should investigate." The following table summarizes the findings of those evaluations, which are included in a physical exam, x-rays, magnetic resonance imaging, which is an MRI, and neurological tests. Okay, so this is our summary of diagnostic testing for Mike Smith. So they've done sensory testing. Okay, so sensory testing is that the information that's coming into the body. Okay, so sensation to touch. So do I know that somebody is touching me? Decreased temperature discrimination. So if I put something cold on them, do they know that it's cold? Or if I put something warm on them, can they feel that it's warm? Okay, so that's that information coming in. Okay, and then they did motor testing, and motor testing is for that information going back out. Okay, so it's strength and movement. Okay, so are they able to move? That is the sense going out. Okay, they did a general examination. That means that they looked at all of the rest of the body, including their pupil response to light. Okay, and so in this case, they had an abnormal pupil response on the right eye. Okay, they did cognitive testing. So cognitive testing is, are you aware of what's going on around you? Do you know what time it is, what place you're in? Okay, are you oriented to the time, the place, and who you are? Okay, so one of them was to count backward from 100 by sevens. So I don't know if you guys can do that, but like 93, 86, 79, 72, right? So if you can do that. All right, and then they also did x-rays and MRI. So they didn't see any fractures in the skull, but they did see a fracture in the 7th cervical vertebrae. Okay, there was significant swelling in the spinal canal from the C7 through the T2 region. So that's the 7th cervical spine, and then down to the 2nd thoracic. Okay, and the spinal cord appeared to be intact, so no injuries there. So now we need to answer some questions. Okay, so based upon those findings that were presented, which doctor made the correct initial prediction? Based upon previous knowledge of brain function, what results from the testing were consistent with the brain injury? And based upon previous knowledge of spinal cord function, what results from the testing were consistent with the spinal cord injury? And based upon your previous knowledge of CNS function, what results would be consistent with both types of injury? Assuming the incorrect diagnosis is true, predict how the findings would be different. Okay, so that's going to be your discussion posts for this week. So for the nervous system, okay, so let's talk about the anatomical divisions. Remember, we talked about how we can divide both anatomically but also functionally. Okay, so we talk about the anatomical divisions. We have the central nervous system. The central nervous system is the brain and the spinal cord, those that are in the center. The peripheral nervous system is everywhere else. All of these are peripheral nerves. Okay. Then we also have functional divisions, okay? So we have the sensation. Remember that sensation is what's happening on the outside. Integration is how do we process it. And then the response, okay, is what are we going to do about it? And then we also have somatic and autonomic. So somatic, that's the things that we're aware of. Autonomic, we're not, okay? So this is just a diagram to kind of show you what's happening. So this is the central nervous system. Okay, here's our brain and our spinal cord. Okay, the brain and the spinal cord are going to send out information. They send it out through the efferent division. Okay, so when we talk about efferent, that means exiting. Okay, so we send that information out efferent. Okay, that efferent division is going to be divided into both the somatic and the autonomic. The somatic, again, that is what we are aware of, what we consciously are controlling. Okay, conscious control here. And so in our bodies, our conscious control is going to be over our skeletal muscles. Okay. We also have our unconscious control. That's our autonomic nervous system. The autonomic nervous system is divided further into two different divisions. We have the sympathetic and the parasympathetic. Okay. Sympathetic, that's going to be your fight or flight response. And parasympathetic, we abbreviate it as rest and digest. Okay, and so what they do is that they send output to the smooth muscles, the cardiac muscles, the exocrine glands, and some of our endocrine glands, right? So this is where the effect is happening. But we also have stimulus coming back. We send that back in through the afferent division, okay? So we have visceral stimuli coming in through our organs, and then we have sensory stimuli coming in through our 5 senses, okay? So what are your 5 senses again? There's sight, smell, taste, hearing, and touch. Okay, so think back to like elementary school and you learned that, right? And so that's our sensory stimuli, pulling that information back in through the afferent, because afferent can say is arriving. Okay, so we have that arriving information coming back into our brain and spinal cord so that we can then do something about it. Okay, so this is our sensory information. We can integrate and then we can respond. Okay, so sensory, integration, response. Okay, so here's our functional divisions. Okay, we also have autonomic and somatic. Okay, also for our functional divisions. Okay, anatomically, we have our central nervous system, that's in the pink, and then we have our peripheral nervous system in the yellow, okay? So there's different cells within our nervous system. We have glial cells. Glial cells are supportive cells. Their job is to support and protect the neurons. And then the neurons are the actual nerve cells. Those are the ones that are receiving the stimulus and then sending out an action potential. Now, remember, we already talked about action potentials, and we're going to talk about it a little bit more, okay? Now, neurons are organized in that they have a cell body. That cell body we also call a soma, okay? We have dendrites, and the dendrites are where we're receiving the information. That's our input. And then we have our axon, and the axon is where we're sending the information. That's our output. Now, we have both gray matter and white matter, okay? And so the gray matter, okay, which you can see right here, there's the gray matter, there's the white matter, okay? And so it's defined by sort of what color is it, right? And so gray and white. So the gray matter, the reason that it appears gray is because it is essentially a group of those neuron cell bodies, okay? Those somas, okay? So all of this is the somas, soma, okay? In the central nervous system, within the brain and the spinal cord, we call a group of those neuron cell bodies, those somas, a nucleus, okay? I know it's confusing, right? Because we've already talked about a nucleus. We've talked about it 2 different times. We talked about the nucleus of an atom. We talked about the nucleus of a cell, right? And now we're talking about it in a whole different way, okay? But this is the nucleus of the central nervous system, okay? In the peripheral nervous system, we don't call it a nucleus. Instead, we call it a ganglion. So if we had a picture of that, of this spinal cord, okay, we would still be calling it a nucleus. If we had a picture of a nerve, the peripheral nervous system, we would call it a ganglion. Okay. Now, within the white matter, we have a bundle of axons. Okay, axons. The reason that they appear white is because if you remember when we originally talked about neurons, is that neurons, that axon, they're covered by this insulation called the myelin sheath. And that myelin sheath is sort of almost has like a fatty consistency to it because it's there for insulation and protection. Okay, and that appears white. Okay, and so within the central nervous system, we call this a tract. And in the peripheral nervous system, we call it a nerve. Okay. What's happening? Let's go back. All right. Apologize. All right, let's see if I can get this back going. Okay. Okay, so there we go. So that gray matter is that group of neuron cell bodies. It's a nucleus in the central nervous system and a ganglion in the peripheral nervous system. And then the white matter is a bundle of axons. And so we call it a tract within the central nervous system and a nerve within the peripheral nervous system. So a nerve versus tract. So we can see this really well when we talk about our optic nerve, okay? And so right here, this is our eye, right? And so we have these optic nerve that's coming in, okay? And then those optic nerves, the optic nerve, remember, is cranial nerve number 1, okay? Number 2, sorry, olfactory is number 1. When we cross here at the optic chiasma, we then become a tract because now we're part of the central nervous system and not part of the peripheral nervous system, okay? So nerve in the peripheral, tract in the central. Same thing, it just has to do with the location, okay? Same thing, it's just a location. It would be like, and I'll share a little secret with you guys, but everywhere in my life, basically, I am called Melissa. However, I grew up in North Carolina, and when I go back to see my grandma or my aunts and uncles, they all call me Missy. So same person, different name depending on where I am. Okay, same idea here. Same stuff, just a different name depending on where it is. So out here, nerve, in here, tract. Okay. So we have some basic functions of our nervous system, right? We have sensory. So those 5 senses that we talked about. Our motor response. Are we going to contract our muscles? Are we going to release hormones? How are we going to change our temperature? And then integration. Integration is where we have the cognition. That's the thinking, learning, forming associations, and processing. Okay. And so with our response division, okay, we have our somatic. Okay. This is conscious and voluntary. So we are consciously perceiving what's going on around us. We can consciously contract our skeletal muscles. In the autonomic, Okay, this is involuntary, and the autonomic nervous system's job is to maintain homeostasis. We find this in both the peripheral nervous system and the central nervous system. This regulates our autonomic functions, right? So our breathing, a release of hormones, digestion, right? And then we move into the enteric. Enteric is considered our digestive nervous system, and it's only found within our peripheral nervous system. It controls our smooth muscle and our glandular tissue in the digestive system. This is the neuron, okay? When we look at that neuron, okay, you can see these are the dendrites. The dendrites are where we're going to have that input. That's where we're receiving the information. You can see there is another neuron right there attached into it, okay? And then we send the information down through what's called the axon hillock and down the axon. That axon is wrapped up in the myelin sheath for protection. It also allows the signal to travel faster. Okay? And then we hit the axon terminals and we connect into another neuron axon to the dendrites at a synapse. We can also, instead of attaching into another neuron, remember we can bind into muscular tissue. Okay. So there's different types of neurons. There's unipolar neurons. Okay, this is called a unipolar. Okay, it's a sensory cell. It's found within the ganglia. Okay, with us in humans, we have what are called pseudo-unipolar. Those are found in mammals because we don't have any true unipolar cells. Bipolar here is uncommon. They're found in our olfactory senses, like with our nose, and then in our retina within our eye. We have multipolar. This is all other neurons. There's also a possible fourth type. They're still doing research on this, and it's where the dendrites can act as the axons. So the dendrites can actually receive and send. They would be smaller and shorter than those other types, but we're still working on whether they are truly neurons or not. Okay, so the typical motor neuron is that multipolar neuron. Okay, so we have, this is our typical one, right? This is a motor neuron, okay? And it's attaching in at that neuromuscular junction into a muscle, right? We also have the Purkinje cells. Purkinje cells are found in our cerebellar cortex right here. And then we have pyramidal cells, and pyramidal cells are found in our cerebral cortex, okay? The cortex, remember, is that outer area. These are our supportive cells. So remember I told you that we have these neuroglial cells, and neuroglial cells are supportive cells. We have different types, whether we're in the central nervous system, brain or spinal cord, or if we're in the peripheral nervous system, all the rest, like our nerves, okay? And so here in our peripheral nervous system, we have satellite cells. Those satellite cells, they're supportive for the neurons, okay? We also have the Schwann cells. So the Schwann cells are going to form the myelin sheath along our peripheral nervous system neurons, okay? In our central nervous system, we have oligodendrocytes. Those oligodendrocytes are doing the same thing as the Schwann cells, except they're in the central nervous system. Their big job is to insulate and form that myelin sheath. Okay. We also have microglia. Microglia are modified immune cells, and they act as scavengers. Okay. And we have astrocytes. Astrocytes are named astrocytes because they are star-shaped. They are sort of taking up all that extra potassium, neurotransmitters. Okay. And then we have our epidermal cells. Okay. The epidermal cells actually help to create barriers between compartments. So these astrocytes, they're star-shaped. They help to form the blood-brain barrier. So most substances can't pass through that blood-brain barrier. Remember, we have cerebral spinal fluid all around our brain and down our spinal cord. We don't have necessarily blood there. We just have that cerebral spinal fluid, CSF. And that is very specific to our nervous system. Those oligodendrocytes, remember, those are going to insulate the axons, and they help to form the myelin sheath. The microglia cells are smaller. They act like macrophages of the immune system. They can eat diseased and damaged nervous system cells. We have the ependymal cells. The ependymal cells help to filter the blood to make that blood-brain barrier, okay? And they line the ventricles in the brain, forming that choroid plexus. And then in the peripheral nervous system, we have those satellite cells. These are similar to astrocytes. They can react to tissue damage. They remove any excess signaling molecules and maintain the concentration. And then we have the Schwann cells right here. Those Schwann cells are just like those oligodendrocytes, but again, they're in the peripheral nervous system, not in the central nervous system. They help to form that myelin sheath. So the myelin sheath is lipid rich. Okay, so that means it's got a lot of fat in it, and it wraps around the axon sort of like a pig in a blanket kind of idea. If you've ever done that, where you're taking the hot dogs and sort of wrapped it up in the crescent rolls or what have you, right? And so it forms insulation. So we get these projections coming out of either the oligodendrocytes or the Schwann cells, and they start to wrap around and sort of grab it, and then it keeps growing around and around and around. And what it does is that it forms this myelin sheath. So there's myelin, and then we have sort of a blank spot, and then another myelin. And between each of these myelin coverings, we have what's called the node of Ranvier. Node of Ranvier. And what this does is that it allows for what's called saltatory conduction. Okay, and saltatory conduction allows for the action potential to jump and move further. So instead of having to travel through, it can actually jump over and over and over and over and over. Okay, and otherwise it would just have to travel along that axon. Okay, and that's called continuous conduction. We do that on an unmyelinated axon. Okay, so just a quick review. Remember we talked about cell membrane proteins. Okay, and so channel membranes are going to allow those ions to pass through, and they do that through diffusion. Okay, remember we moved from an area of high concentration to an area of low concentration. Okay, we also have ligand-gated channels. Ligand-gated channels are where we have a neurotransmitter or something else bind to the channel, and that opens it. It's like the key. Okay, that ligand is like a key. And so we put the key in this case, it's acetylcholine. And when that happens, we can open up the channel. And then we have mechanically gated channels. Okay, those mechanically gated channels are actually the door, and a door has to physically open. Okay. And then we have voltage gated channels. So depending on what the voltage is, okay, is that we can open it. So you can see it's sort of this voltage gated channel down here. It's closed. Once the voltage hits a certain point, the ball falls out, okay, and then we can open that door. And then we have leakage channels. Okay, leakage channels are randomly opening and closing. So what happens when our pathway gets activated? Okay, so we have sensory endings within our skin, right? One of our senses is touch. Okay, and one of them with the touch is about temperature, right? And so here we're putting our hand under some water, okay? And that's then going to send an action potential through our sensory axons, right? So this is an arriving signal, so this is an afferent pathway, okay? And that sensory axon is going to enter the spinal cord, and it synapses with the brain through what's called an interneuron. Interneuron means between neurons. Okay, so this is that sensory neuron, afferent neuron, entering into that interneuron. And then we're going to send it through a second neuron projecting with the thalamus. Okay, that's going to reach the cerebral cortex. We do our conscious perception here at the cerebral cortex. Okay. Once we do that, we're going to send a message through our upper motor neuron. The upper motor neuron from the cortex is going to execute that motor command. Okay. And it's going to contact a lower motor neuron within the spinal cord. And this would be an efferent pathway here. Okay, to that lower motor neuron, okay, that's going to cause a contraction of our target skeletal muscles. Okay. So let's review action potential. Okay, so what's happening in each number here, right? We get a stimulus. So that stimulus is that we have activated that ligand-gated channel, right? And so when we activate that ligand-gated channel, that's going to start the depolarization of the cell. Once we hit threshold of negative 55 millivolts, we're then going to open up our voltage-gated sodium channels. Remember, sodium is flooding in into that cell, because that cell started out as okay, and the sodium was on the outside, okay, but it was very negative on the inside, right? So when we get that stimulus of that ligand binding to a receptor, like the acetylcholine binding to the receptor, okay, when that happens, again, we're going to open up those sodium channels and sodium is going to flood in. Once we hit threshold of about negative 55 millivolts, we're going to open up those voltage-gated sodium channels, and more sodium is flooding in, flooding in, and flooding in. Okay. Then when we hit our peak, okay, those voltage-gated sodium channels are going to close. Okay. When that happens, we're also going to open up our voltage-gated potassium channels. And now, because it's so positive on the inside of that cell, because we allowed all that positive sodium in, now the potassium is going to flood out, because it's like, let me out of here. It's too positive. It's too happy in here, so let me out. Okay. Then that potassium is going to flood out, and it continues to flood out until those voltage-gated potassium channels close. Okay. And then our sodium potassium pump is going to pump us back to normal and put us back at our resting state. Okay. So we have what are called absolute and relative refractory periods. During an absolute refractory period, another action potential cannot start. During what we call a relative refractory period, another action potential can start. Okay, but the stimulus will have to be stronger. We also have what are called graded potentials. This is where we have a smaller stimulus causing a small depolarization, but we don't hit threshold. Okay, so once we hit threshold, that can trigger that action potential. We also have some stimuli that result in hyperpolarization. So that means that the stimulus would have to be even stronger to make us do an action potential. Look, we're hyperpolarized here, right? So we're even further than that negative 70, okay? And that would mean that we'd have to allow even more sodium in to hit threshold, right? So it would cause us more time usually, okay? Now remember the synapse. This is where we have a connection between a neuron and the target cell. That target cell can be a neuron, but it doesn't have to be another neuron. We can think back to that neuromuscular junction where, remember, we had muscles here, okay? But in this case, we have two neurons. We have a presynaptic neuron. Presynaptic means that it's before the synapse. And then we have a postsynaptic neuron, okay? So this would be the dendrites. This is the axon terminal, okay? So the presynaptic element is the synaptic end bulb here. That's where calcium is going to enter, and it causes these vesicles to diffuse or to release, okay, those neurotransmitters. Those neurotransmitters are going to diffuse across the synaptic cleft. They're going to bind to those receptors over here, and they open up those ligand-gated channels. Once we've finished and we're no longer releasing, okay, because the stimulus has stopped, okay, we can do enzymatic degradation, neuronal reuptake, meaning that the neurons are going to reuptake it, or glial reuptake. So the glial cells are going to pull back those neurotransmitters, okay. There's different types of neurotransmitter systems. There's the cholinergic system, amino acid systems, biogenic amine systems, and neuropeptide systems. Okay. So the cholinergic system is that we have the neurotransmitter acetylcholine. And there's two different types of receptors. Okay. There's nicotinic and muscarinic. Okay. Nicotinic and muscarinic. In this case, when we release that acetylcholine, we're going to eliminate it by the degradation of acetylcholinesterase, which is an enzyme to break down acetylcholine. Okay. And the postsynaptic effect means what's going to happen on the next cell. Okay. It can either be excitatory or inhibitory. Excitatory or inhibitory. Sometimes it can even be both. Okay. And it depends on the receptor. The receptor is what tells us. So we're releasing acetylcholine. That's our neurotransmitter. But what is it going to do depends on the receptor. Okay. In the amino acid systems, this is our most abundant class, and they function only within our central nervous system. Okay, there's different neurotransmitters. There's excitatory like glutamate and aspartate. Aspartate might sound similar to aspartame. Okay, there's also inhibitories, right? Glycine and GABA. And then we have the different receptors. The receptors are named for the neurotransmitters. So we have GLU receptors, GLI receptors, and GABA receptors. They're going to be eliminated because they're going to be reuptaked by the neurons or by those glial cells. And they can be excitatory with the GLU receptors, or they can be inhibitory with the GLI or the GABA receptors. Okay, so excitatory, when we talk about it being excitatory, that means it's going to cause that depolarization. We're going to start heading up towards threshold. For inhibitory, it means we're going to be hyperpolarized. So we're going to go down instead of up in our curve. For the biogenic amine systems, we have different neurotransmitters. We have serotonin. Serotonin helps us to regulate our sleep and emotions. We have dopamine and norepinephrine, and also epinephrine, helps to regulate our autonomic nervous system motor functions. And then histamine. Histamine causes us an inflammatory response. You would take an antihistamine when you're having an allergic response, okay? And so the histamine causes that inflammation. There's different types of receptors, okay? And we eliminate them by reuptake by those neurons, okay? And so the postsynaptic effect depends on the specific receptor, just like all the other ones, right? So D1 receptors can cause depolarization, while D2 receptors cause hyperpolarization. Okay. Then we have the neuropeptide systems. These are neurotransmitters like metenkephalin, beta endorphins, VIP, substance P. Again, the receptors are specific to the peptides. We degrade these by enzymes called peptidases, and the postsynaptic effect depends on what receptor, just like all of the other ones, okay? Sometimes we consider these neuromodulators because they can alter the response or the action of a neurotransmitter, and so we can call them hormones. For instance, TRH, which is thyroid-releasing hormone, causes the release of TSH, thyroid stimulating hormone. Substance P decreases our gastrointestinal motility. Vasopressin regulates our urine output by the kidneys. And endogenous opiates give us an analgesic effect or pain relief and a euphoric response. This is a chart that I would like you guys to fill out. That is going to be your next lab assignment. Okay. Or not lab assignment, your next assignment for lecture. Okay, and all right, guys, that is everything for today. If you have any questions, please reach out to me and I will talk to you guys soon.